Filopodia. 10 languages. This electron micrograph shows exaggerated Filopodia with club-like shape induced by formin MDA2 in cultured cells. These Filopodia are filled with bundled actin filaments, which were born in and converged from the lamellipodial network. Microspikes redirects here. For the hiking footgear, see crampons microspikes. Filopodia, SG, Filopodium, are slender cytoplasmic projections that extend beyond the leading edge of lamellipodia in migrating cells. 1. Within the lamellipodium, actin ribs are known as microspikes, and when they extend beyond the lamellipodia, they are known as Filopodia. 2. They contain microfilaments, also called actin filaments, cross-linked into bundles by actin bundling. Proteins, 3, such as fascin and fibrin. 4. Filopodia form focal adhesions with the substratum, linking them to the cell surface. 5. Many types of migrating cells display Filopodia, which are thought to be involved in both sensation of chemotropic cues, and resulting changes in directed locomotion. Activation of the Rho family of GT parses, particularly CDC42 and their downstream intermediates, results in the polymerization of actin fibers by NA/VASP homology proteins. 6. Growth factors bind to receptor tyrosine kinases, resulting in the polymerization of actin filaments, which, when cross-linked, make up the supporting cytoskeletal elements of Filopodia. Rho activity also results in activation by phosphorylation of esrin-moesin radixin family proteins that link actin filaments to the Filopodia membrane. 6. Filopodia have roles in sensing, migration, neurite outgrowth, and cell-cell interaction. 1. Further explanation needed. To close a wound in vertebrates, growth factors stimulate the formation of Filopodia in fibroblasts to direct fibroblast migration and wound closure. 7. In macrophages, Filopodia act as phagocytic tentacles, pulling bound objects towards the cell for phagocytosis. 8. Functions and variants, edit. Many cell types have Filopodia. Citation needed. The functions of Filopodia have been attributed to pathfinding of neurons. 9. Early stages of synapse formation. 10. Antigen presentation by dendritic cells of the immune system. 11. Force generation by macrophages. 12. And virus transmission. 13. They have been associated with wound closure. 14. Dorsal closure of Drosophila embryos. 15. Chemotaxis in dictyostelium. 16. Delta notch signaling. 17. 18. Vasculogenesis. 19. Cell adhesion. 20. Cell migration, and frequently asked questions. Other ways to give. I already donated. How often would you like to donate? One time give monthly. Please select an amount, USD. The average donation in the United States is around $13. $2.75, $10, $15, $25, $50, $75, $100 other. Maybe later. 1. Suzo RCNA. 2. Next, choose payment method. Wikipedia still can't be sold. The 17th of October, an important update for readers in the United States. You deserve an explanation, so please don't skip this 1-minute read. We're sorry to interrupt, but it's Thursday, the the 17th of October, and this message will be up for only a few hours. We ask you to react on the number of times you visited Wikipedia in the past year, and if you're able to give $2.75 to the Wikimedia Foundation. If everyone reading this gave just $2.75, we'd hit our goal in a few hours. In the age of AI, access to variable facts is crucial. Wikipedia is at the heart of online information, powering everything from your personal searches to emerging AI technologies. Your gift strengthens the knowledge of today and tomorrow. Just 2% of our readers donate, so if you have given in the past and Wikipedia still provides you with $2.75 worth of knowledge, kindly donate today. If you are undecided, remember that any contribution helps, whether it's $2.75 or $25. Proud host of Wikipedia and its sister sites. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Cancer metastasis. Specific kinds of Filopodia have been given various names: citation needed, microspikes, pseudopods, thin Filopodia, 21, thick Filopodia, 22, gliopodia, 23, myopodia, 24, invadopodia, 25, podosums, 26, telepods, 27, tunneling nanotubes, 28, and dendrites. In infections, edit. Filopodia are also used for movement of bacteria between cells, so as to evade the host immune system. The intracellular bacteria, Ehrlichia, are transported between cells through the host cell Filopodia induced by the pathogen during initial stages of infection. 29. Filopodia are the initial contact that human retinal pigment epithelial (RPE) cells make with elementary bodies of chlamydia trachomatis, the bacteria that causes chlamydia. 30. Viruses have been shown to be transported along Filopodia toward the cell body, leading to cell infection. 31. Directed transport of receptor-bound epidermal growth factor, EGF, along Filopodia has also been described, supporting the proposed sensing function of Filopodia. 32. SARS-CoV-2, the strain of coronavirus responsible for COVID-19, produces Filopodia in infected cells. 33. In brain cells, edit. This section needs expansion. You can help by adding to it. February 2023. In developing neurons, Filopodia extend from the growth cone at the leading edge. In neurons deprived of Filopodia by partial inhibition of actin filaments polymerization, growth cone extension continues as normal, but direction of growth is disrupted and highly irregular. 7. Filopodia-like projections have also been linked to dendrite creation when new synapses are formed in the brain. 34, 35. A study deploying protein imaging of adult mice showed that Filopodia in the explored regions were by an order of magnitude more abundant than previously believed, comprising about 30% of all dendritic protrusions. At their tips, they contain silent synapses that are inactive until recruited as part of neural plasticity and flexible learning or memories, previously thought to be present mainly in the developing pre-adult brain and to die off with time. 36, 37, further explanation needed. References, edit. 1 AB Matala Park, Lapalainen P. June, 2008. Filopodia. Molecular architecture and cellular functions. Nature. Reviews. Molecular cell biology. 9.6446454. Do. 10.1038 / NRM 2406. PMID 1846470. S2 SID 335333182. 2 small JV, Stradal T, Vinyl E, Rotner K, March 2002. The Lamellipodium, Where Motility Begins. Trends in Cell Biology. 12. 31122120 10.1016 /S0962-8924 01 02237-1. PMID 118590023. 3 Kurana S., George S. P., September 2011. The role of actin. Bundling proteins in the assembly of Filopodia in epithelial cells. Cell adhesion and migration. 5.5409-420. Do. 10.4161 /cam.5.5.17644. PMC 3218608. PMID 2197550. 4 Hanine D., Matsudera P., DeRosier DJ, October 1997. Evidence for a conformational change in actin induced by fibrin, N375, binding. The Journal of Cell Biology. 139, 2. 387396. Do. 10.1083 /JCB.139.2.387. PMC 2139807. PMID 9334343. 5 Lodish H., Burko, Matsudera P., Kaiser C.A., Krieger M., Scott M.P., Zipersky S.L., Don L.J., eds. 2004. Molecular Cell Biology. 5th ed. W.H. Freeman and Company. pp. 821, 823. 6 AB Otter Y, Suzuki N, Nakamura S, Hartwig J.H., Stazel T.P. March 1999. The small GT passe relay targets filamen to induce Filopodia. Proceedings of the National Academy of Sciences of the United States of America. 96, 5 2122 2128. Bibcode 1999 NAS 96.2122. Do. 10.1073 /NAS.96.5.2122. PMC 26747. PMID 10051605. 7 AB Bentley D., Toroyan Raymond O., 1986. Disoriented. Pathfinding by pioneer neuron growth cones deprived of Filopodia by cytocalysin treatment. Nature. 323, 6097 12. 715 Bibcode, 1986 Nature.323712b. Do. 10.1038 /3237120. PMID 3773996. S2 SID 4371667. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 8 Cress H, Stiza A, Holzer D, Bus F, Griffiths G, Rohrbach. July 2007. Filopodia act as phagocytic tentacles and pull with discrete steps and a load-dependent velocity. Proceedings of the National Academy of Sciences of the United States of America. 104, 281163311638. Bib code 2007 NAS 104116333K. Do 10.1000/NAS.0702449104. PMC 1913848. PMID 176200618. 9 Bentley D. Toroy and Raymond O. 1986. Disoriented. Pathfinding by pioneer neuron growth cones deprived of Filopodia by cytocalysin treatment. Nature. 323, 6097 125. Bib code 1986 Nature.323712b. Do 10.1038/323712A0. PMID 3773996. S2 SID 4371667. 10 Yustar, Bonhoeffer T., January 2004. Genesis of dendritic spines, insights from ultrastructural and imaging studies. Nature Reviews. Neuroscience. 5, 12434. Do 10.1038/NRN 1300. PMID 147080001. S2 SID 151262232. 11 Raghunathan, Sivakamarjandriya, Walensky J., Podawa. Weissman S.M., August 2001. Functional Analysis of B144/LST1, a gene in the tumor necrosis factor cluster that induces formation of long filopodia in eukaryotic cells. Experimental Cell Research. 268, 223044. Do 10.1006/xk.2001.5290. PMID 114788849. 12 Cress H, Stiza A, Holzer D, Bus F, Griffiths G, Rohrbach A, July 2007. Filopodia act as phagocytic tentacles and pull with discrete steps and a load-dependent velocity. Proceedings of the National Academy of Sciences of the United States of America. 104, 28116338. Bib code 2007 NAS 104116333K. Do 10.1038/NAS.0702449104. PMC 1913848. PMID 176200618. 13 Learman M.J., Shearer N.M., Marks C.B., Pipert M., Moths W. July 2005. Actin and myosin-driven movement of viruses. A long filopodia precedes their entry into cells. The Journal of Cell Biology. 170, 231725 do 10.1083/JCB.2005033059. PMC 2171413. PMID 160272225. 14 Crossan CE, Kleiss SD, Buermann RW, April 1986. Epithelial wound closure in the rabbit cornea. A biphasic process. Investigative Ophthalmology and Visual Science. 27. 446,473. PMID 3957565. 15 carat Jacintua, Wood W, Barley OT, Termain M, Martin S. Arias A. Martin P., November, 2000. Dynamic actin-based epithelial adhesion and cell matching during Drosophila dorsal closure. Current biology. 10.22.1426. Bib code 2000 CBio 10.1420J. Do 10.1016/S0960 9822 00000796X. PMID 11102803. 16 carat Han Ye, Chung CY, Wessels D, Stevens S, Titus Ma, Sol. Dr. Fertile Ra, December, 2002. Requirement of a vasodilator. Stimulated phosphoprotein family member for cell adhesion, the formation of Filopodia, and chemotaxis in dictyostelium. The Journal of Biological Chemistry. 277, 51498777. Do 10.1074/JBC.M2091072000. PMID 123888544. 17 carat Cohen M, Georgiou M, Stevenson NL, Mayo Downique M, Baum B. July 2010. Dynamic Filopodia transmit intermittent delta notch signaling to drive pattern refinement during lateral inhibition. Developmental cell. 19, 17889. Do 10.1016/J.Devsel.2010.06.006. PMID 206433552. 18 carat Berkemeier F, page, kilometer, June 2023. Coupling dynamics of 2D notch delta signaling. Mathematical biosciences. 360. 1. Do 10.1016/J.MBS.2023.109012. PMID 37142213. 19 carat Lawson ND, Weinstein BM, August 2002. In vivo imaging of embryonic vascular development using transgenic zebrafish. Developmental biology. 248, 230718. Do 10.1064/bio.2002.0711. PMID 121674006. 20 carat Vajak Hin V, Bauer C, Yun M, Fuchs C, January 2000. Directed actin polymerization is the driving force for epithelial cell-cell adhesion. Cell 100, 220919 Do 10.1016/S0092. 8674, 00, 81559-7. PMID 106600044. 21 carat Miller J, Fraser S.E., McClay D., August 1995. Dynamics of thin Filopodia during sea urchin gastrulation. Development. 121, 82501 Do 10.1242/dev.121.8.2501. PMID 7671814. 22 carat McClay Dr., December 1999. The role of thin Filopodia in Motility and morphogenesis. Experimental cell research. 253. 2296301 10.1006.1999.4723. PMID 1058550. 23 Vazenkova I, Lujimbaldi, Chibara, January 2006. Gliapodia. Extend the range of direct glia-neuron communication during the CNS development in Drosophila. Molecular and cellular neurosciences. 31, 112330. Do. 10.1016/j.mcn.2005.10.001. PMID 1629814. S2 SID 39541898. 24 Ritson-Thaler S, Suzuki E, Chibara, October 2000. Postsynaptic Filopodia in muscle cells interact with innervating motoneuron axons. Nature Neuroscience. 3, 10.1012-7. Do. 10.1038/79833. PMID 110171774. S2 SID 237188828. 25 Chen Wait, August 1989. Proteolytic activity of specialized surface protrusions formed at rosette contact sites of transformed cells. The Journal of Experimental Zoology. 251. 216785 do. 10.1002/jes.14025100206. PMID 2549171. 26 Taran G, Cirillo D, Giancotti FG, Comaglio PM, Marchizio PC. July 1985. Sarcoma virus transformed fibroblasts. Adhere primarily at discrete protrusions of the ventral membrane called podosomes. Experimental cell research. 159, 114157 do. 10.1016/S0014-4827, 85, 80044-6. PMID 2411576. 27 Popescu LM, Fausen Pellegrini MS, April 2010. Telocites, A Case of Serendipity, The Winding Way from Interstitial Cells of Kagel, ICC, via interstitial Kagel-like cells. ICLC, to telocites. Journal of Cellular and Molecular Medicine. 14, 472940 do. 10.1011/j.1582. 4930.2010.01059.x. PMC 38231108. PMID 203676664. 28 Rustomer, Safrika, Markovic I, Walther P., Gerds H.H. February 2004. Nanotubular highways for intercellular organelle transport. Science. 303, 5661,00710. Bibcode, 2004 SCI, 303.1007R. Do. 10.1116/science.10933133. PMID 149633329. S2 SID 37863055. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 29 Thomas S., Popov VL, Walker DH, December 2010. Exit. Mechanisms of the intracellular bacteria Maiolichia. PLOS 1. 5, 12E15775. Bibcode 2010 PLOSO 515775T. Do 10.1371/journal.pwn.0015775. PMC 3004962. PMID 2118797. 30 Ford C, Nanzo, Bucrotti, Hayward Road, May 2018. Welsh. MD, ED. Chlamydia exploits filopodile capture and a macropinocytosis-like pathway for host cell entry. PLOS. Pathogens. 14, 5E10070551. Do 10.1371/journal.pat.10070551. PMC 5955597. PMID 297274663. 31 Learman MJ, Shearer NM, Marks CB, Piperet M, Moths W. July 2005. Actin and myosin-driven movement of viruses. A long Filopodia precedes their entry into cells. The Journal of Cell Biology. 170, 2317325 do. 10.1083/jcb.200503059. PMC 2171413. PMID 160272225. 32 Lidg DS, Lidg KA, Riga B, Jovin TM, and Jovin DJ. August 2005. Reaching out for signals, Filopodia sense EGF. And respond by directed retrograde transport of activated receptors. The Journal of Cell Biology. 170, 4619626. Do 10.1083/jcb.200503140. PMC 2171515. PMID 161032229. 33 Buhadu M, Memon D, Maya B, White Kilometer, Rizelj VV. Korea Mauro M, it al. August 2020. The Global Phosphorylation Landscape of SARS-CoV-2 Infection. Cell 182, 3685712.E19 do 10.1016/j.cell.2020.06.034. PMC 7321036. PMID 326455325. 34 Beardsley J, June 1999. Getting Wired. Scientific American. 280, 624. Bibcode 1999 SCIM.280F24B. Do 10.1038/Scientific-American 0699-24B, inactive 2024-09. 25. 35 Maletic Savatic M, Malina R, Svoboda K, March 1999. Rapid dendritic morphogenesis in CA1 hippocampal dendrites. Induced by synaptic activity. Science. 283, 5409 1923 1927. Do 10.1126/science.283.5409.1923. PMID 10082466. 36 Lauder, Claudia Lopez, the 16th of December 2022. Adult mouse. Brains are teeming with silent synapses. Retrieved the 18th of December 2022. 37 Vardalaki, Dimitra, Chang, Kwanghan, Harnett, Mark T. December 2022. Filopodia are a structural substrate for silent synapses in adult neocortex. Nature. 612, 7939 323327. Bibcode 2022 nature.612323v. Do 10.1038/S41586-022. 05483-6. ISSN 1476-4687. PMID 364509884. S2 SID 2541222483. University Press Release, Trafton, Anne. Silent synapses are abundant in the adult brain. Massachusetts Institute of Technology via medicalexpress.com. Retrieved the 18th of December 2022. External links, edit. MB Info, Filopodia. MB Info, Filopodia Assembly. New form of cinema, cellular film, proposal for documentaries with cellular imaging. Categories: cell movement, cytoskeleton, cell biology, neurons, actin-based structures. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.